Thanks, Pauline. Our next speaker is Stephen Mount.
Okay, so happy to be here. I'm going to tell you today a very small story about very small exons called nano-exons. This is primarily the work of Yufei Shi, a graduate student in my group, and we've taken advantage of a very nice data set from colleagues Louisa Wu and the graduate student Ashley Nazario-Toole. I can't see the... Anyway. Is the mouse working? I can't see the arrow.
It's not advancing. Is there anything you can do at the back?
Is the mouse unplugged or something? Okay, so I'm going to talk to you today about what nano-exons are. I'll briefly review. Okay, advance the slide. I have lots of slides, so I have to be able to advance them. Okay, I'm going to review exon definition, micro-exons, resplicing sites. I'm going to define nano-exons as exons less than 4 nucleotides, and talk about how small that can be. I'll argue that there can be exons of size -1, and I'll tell you what I mean by that. I'll tell you how we recognize nano-exons, which is or how we detect them by aligning RNA-seq reads to the transcriptome, not the genome, and the background rate of, this works because the background rate of Indel variants or mutations or errors in RNA-seq data is very low. Next slide. So we have found some exons of size -1 and some other nano-exons, and I'll be describing them. Next slide. So exon definition, how are small exons recognized in general? The idea of exon definition was proposed by Sue Burgett many years ago, and it depends on the cooperative interactions. I can't use the mouse. Oh, here it is. No, this is not working. Can I use the pointer? This is ridiculous. There's no pointer. And this is not. Oh, there it goes. Okay, so micro-exons are, in general, small exons are recognized by cooperative binding of components to the two ends of the exon. Factors at the 3' splice site, the 5' end of the exon, and the 5' splice site, the 3' end of the exon bind cooperatively, resulting in the efficient recognition and removal of the exon. The next slide shows the distribution of exon sizes. There's a preferred size of about 100 nucleotides, which reflects this cooperative interaction. Next slide. Micro-exons are defined as, I don't think it's going to, is it even going to show up? Yeah, okay. So, what happened? Oh, no, it went back. Next slide. Is this, okay. Micro-exons are small exons. They've been defined as less than 52 or less than 30. The exact size can vary. Exon sizes vary sort of smoothly down to very small sizes. I tend to think of micro-exons as defined by being too small for cooperative binding between the two ends of the intron. The next slide shows that, and the next slide illustrates how the micro-exons can be removed by sequential removal of first one intron and then the other intron, either the upstream intron first, as shown here, or the next slide, removal of the downstream intron first can also be observed. Okay, so the next slide then shows recursive splicing, which is the removal of an exon of 0 nucleotides, basically by a resplicing at a single site within the intron. And again, this can occur either the upstream intron or the downstream intron first. So thinking about very small exons as being removed in 2 stages, you can then consider the next slide a case of 3-nucleotide AGU, which fits the consensus dinucleotides in the middle of a very long intron, can be removed through 2 steps. And the first step will then place the terminal G, either the downstream exon shown on the left or the upstream exon shown on the right, for removal in a second resplicing step. So the sequence within the long intron containing the AGU sort of compound conserved dinucleotides sets up resplicing to remove a single nucleotide, and we consider this an exon of length -1. Of course, there's no actual exon of size -1, rather there's a sequence within the long intron which acts to remove a single nucleotide. So we were interested in whether we could find such nanoexons, and we looked at all exons of less than size less than size 4. Okay, next slide then gives my current definition of that as a nanoexons less than 4. And bioinformatically, nanoexons can be thought of as exons which are small enough that when reads are aligned to the genome or even to the transcriptome, the gap would simply allow the alignment to occur without the exon sort of causing a problem. So the next slide then shows how we detected nanoexons. We aligned reads not to the genome but to the transcriptome, and then we screened those alignments for indels, insertions or deletions of less than 4 nucleotides that were near an annotated splice site, and then we considered those further by looking for the supporting evidence for that. Okay, so this depends, the next slide, on the background rate of Indel. So the data set we used was a high-quality Illumina RNA-seq data set from Drosophila hemocytes with and without RNAi knockdown of the splicing factor A2BP1, which is the Drosophila homolog of the RB-Fox splicing factors. Next slide then shows the sort of algorithm. We look for indels of less than 4 nucleotides. The gap, there could be no mismatches, and the gap had to be within sort of the center of the read, at least 7 nucleotides away from each end. Okay, so looking at this, the next slide shows us, shows the sort of background rate of indel reads in RNA-seq. These could be due either to errors during reverse transcription, errors during sequencing, or misannotations in the genome. The red line shows you 1 over the total counts, and the black line shows you the distribution of reads with indels, and you can see that there's an extremely low rate of indels of this kind. So we have all positions. The highest rate we saw at any position was 18%. Very few positions showed any indels, and only 38 positions out of a total of, actually, next slide is the number, 49,000 positions showed rates over 1%. Okay, so indel rates are extremely low. Those that do occur almost always occur at runs of A's and T's homopolymer stretches. So next slide, what we did is we took our alignments from the entire RNA-seq data set. We removed indels that occurred at homopolymer runs. We removed indels that we saw only a few times because they were probably just errors. And we ended up with an observed case of 11 previously undescribed nanoexons. Not that many. We also found a large number of closely spaced 5' and 3' splice sites, mostly 3' splice sites fitting the NAG pattern. Okay, so Okay, so there are not very many nanoexons, but there are a few, and I'll just go through a couple of cases in some detail. The first one I want to describe is axon. Next slide shows the, so the three where we found a -1 nanoexon or a removal are axon, orb, and RPL8. Orb did not check out when we looked at modern code data and may simply be a polymorphism, so I really should say we have 2, axon and RPL8. The next slide shows axon in some detail. This is these are all in the 5' UTR. The axon -1 nanoexon is in the first intron of a long 5' UTR. There are highly conserved branch site and AGT resplicing sites within with this intron. And next slide, we looked at modern code data. We found that go back one, we found that the percent spliced out, if you will, is always about 50%, varies from 22% to 67% in the different stages of that data set. The next slide shows RPL8, which is a sort of different case. Here you have a very high expression of a ribosomal protein, 17,000 reads crossing this intron in our data set. There's a minor form where the sort of upstream half of this intron in the 5' UTR is removed. And if you look at that mRNA and resplice it, which occurs at a low rate, you get the -1 nanoexon. So this, again, next slide, is observed in modern code data. It's a variable, but always quite low rate. Next slide. And the next slide. Okay, so this is a case of a small 3-nucleotide exon that includes 1 amino acid, has not been previously observed in the FHOS gene. I'm not sure how you pronounce that. This is a highly conserved protein, conserved site, and this is alternatively spliced. Next slide summarizes that data from the modern code, where the percent spliced in varies from 0 to 90%. And then the final slide, or final data slide, next slide, shows example of a 1-nucleotide exon from a vacuolar sorting protein. This single nucleotide exon is required to maintain the frame of the protein, which is conserved, and had not been described. The UC Santa Cruz, based on the genome annotation, gives the incorrect prematurely terminated protein sequence. Flybase gets it right by reference to an EST sequence, but there's no annotated nucleotide in the genome. Okay, so next slide is my conclusions. We do find nanoexons. There are not very many. They can be recognized by RNA-seq reads mapping to the transcriptome. We find nanoexons affecting a protein sequence, and these are kind of useful in improving annotations. I think more generally, I would recommend that people consider mapping RNA-seq reads to transcriptome, not to genome, and there are methods like Salmon and Callisto which allow that to be done for differential expression. And I guess that's it. Thank you very much.
That's really interesting. I get in my mind the fact that there are these intronic enhancers and suppressors of splicing that look like 0-nucleotide exons. So in other words, a 3' splice site directly adjacent to a 5' splice site. There's one in CTCGFP that I worked on in the last century. I'm wondering if some of these occurrences that you've observed are kind of low frequency consequences of using a regulatory element like that, where you have the machinery there, and then occasionally it triggers a recognition event by the spliceosome when what it's really doing is acting as a regulator of some kind of broader alternative splicing event. I'm sure you can address that.
Well, I guess one thing I would say is there are more resplicing sites than there are nano-exons, right? So if you're just talking about kind of the regulation of splicing at a site where you're going to splice twice, there's evidence for more resplicing sites than nano-exons.
No, something where there's like an element in the middle of the intron that's going to cause alternative terminal exon usage.
Yeah.
But where occasionally, because there's a U1 SNRP and a U2 SNRP sitting there, the spliceosome might, fire, essentially.
Well, what you're talking about is similar to what was originally described in UBX, where you have micro-exons which can either be skipped or included, and when they're skipped, they're actually respliced as nano-exons.
So are the adjacent exons, do they tend to be alternatively spliced? I think that's
Not in the few
question.
cases I showed. No.
Okay.
So on the slide about Haas, or however you pronounce
Yeah.
it, suggested that the splicing that led to the inclusion of the amino acid was in the nervous system. So I was just curious if most of what you're seeing are nervous system events.
The two -1 cases were observed pretty much throughout development and in multiple tissues. I think they were all the tendency for the nervous system to show a different rate. I can't remember the details. Yeah.
Sort of following up on that question, I don't know whether I missed it or not. What are the biological functions of these nano-exons, or do you see any defects, especially in the nervous system? Because these targets are very important for neurodevelopment. Do you see any defects, or like if you don't have these?
Well, so the RPL, the ribosomal protein case, is probably just like noise, because there's a very high rate of expression. You're just resplicing that minor form. The axin case may be regulatory because it is differentially alternatively spliced in tissues. It is in the 5' UTR, and I don't know anything about, I can speculate that maybe it plays some regulatory role, removes or adds a binding site, but we really don't know. In the case of the single amino acid insertion, again, that's all alternatively spliced, and it's likely to affect function, but I don't know much about it.